forest plot is a diagram produced as one of the output from meta-analysis, as shown here in this diagram. This is forest plot. So what is a forest plot? Forest plot is a graph that presents the summary of a meta-analysis results. So it displays the magnitude of the effect size across different studies. So the meta-analysis is a statistical method that will combine data extracted from several independent clinical or research studies that addressing specific question. Those data are combined and analyzed based on the meta-analysis method, and from the method an output is produced, which is a forest plot. So the forest plot will display the effect size across those different studies, and the plot will also identify sources of heterogeneity in the results, as these data are collected from several studies, and the plot will show and determine whether there is heterogeneity and the level of the heterogeneity and the magnitude of the heterogeneity. So the plot, forest plot, consists of three important parts. The first part is descriptive statistic that shows the summary for this descriptive statistic. The second part is a diagrammatic representation of this statistics, and the third part is the statistical analysis summary. So there are three sections. The first section, as shown here, is the descriptive statistics. The second part is the diagrammatic representation of this descriptive statistics, and the third part, which is the statistical analysis, which shows the overall estimate of the effect size and the heterogeneity test results. So the first part is a descriptive statistics, as shown in the plot here generated from SPSS. It shows in the first part the summary for each study. There are 10 independent studies combined, and the effect size was used as the standardized mean difference, which is the Hedges G, and the value is scored here, and the 95% confidence interval is given for the upper and lower limits, and statistical p-value is generated with a weight percentage, weight and a weight percentage. So the first part here is the estimate effect size, which is Hedges, which based on a continuous outcome or continuous variable based on the mean, and the 95% confidence interval is generated, and a p-value for this Hedges is also produced, and a weight percentage. A weight percentage describes and shows these values the size of the sample or the number of participants, and larger samples will have higher weight percentage. So the summary for each study is shown here, and as described, effect size for each study with the sample size, which is not listed here, but it could be added, 95% confidence interval, the weight percentage, and the p-value. So this is the first part of the forest plot, which is descriptive statistics. There are different type of effect size depends on the outcome. If the outcome is continuous, the effect size could be the mean difference or the standardized mean difference, and that could be one of these taken or considered, such as the Hedges G, the Cohens D, or Glacis delta. If the outcome also is continuous, the effect size can be a correlation coefficient R, and if the outcome is binary, which has two levels in it, yes or no, life or dead, then the effect size could be the odds ratio, the risk ratio, the hazard ratio, or proportion. So the forest plot now have three parts. The first part is the descriptive part. The second part is a diagrammatic part or diagrammatic representation of the descriptive statistics. As shown here in this diagram, there are symbols, squares, horizontal line, vertical lines, diamond-shaped structure. This part is a graphical representation of the descriptive statistics. So the square symbol here, the blue color coded squares, are the effect size estimate, and there are different sizes of these squares, and that is a reflection of the sample size. Larger sample size will have larger square, smaller sample size or smaller study with smaller number of participants will have smaller square size. The horizontal line here represent the 95% confidence interval for each specific effect size for each study, and the diamond shape here, or the diamond shaped symbol, represent the overall effect size estimated from pooling all these different effect size from those separate studies. The blue line here is the line of no effect, and the red dashed line here is the line of the overall effect size, which is calculated and found to be 0.53. As I have described previously, the vertical blue line here at a value of 0 on a continuous scale for continuous variable, continuous outcome, is the line of no effect. If the confidence interval for the study effect size estimate overlaps with this line, the 0 line, that suggests that the study results are not statistically significant. The third part of the forest plot is the section here, which is a statistical analysis, which is the overall estimate of the effect size. So it shows the overall or pooled effect size with p-value and 95% confidence interval, and it's shown here also the overall, and the effect size, which is Hedges G, is 0.53, and the 95% confidence interval given with a p-value. And also in this section, the measure of heterogeneity is performed for heterogeneity test, and an I-squared value is calculated with an estimate for the p-value. This measure of heterogeneity indicates heterogeneity in the effect size across studies, and it is calculated based on Cochran's Q-statistics with I-squared value generated and a p-value generated, and a tau squared value. So from this forest plot, the pooled estimate of effect size shows that there is a statistically significant difference between intervention and control group based on the mean for each group, in which it's calculated as standardized mean difference using the Hedges G, and this difference is statistically significant as the p-value is less than 0.05 and the 95% confidence interval contains the Hedges overall Hedges value. That's the for first part of the analysis. The second part shows that there are not high level of heterogeneity among the result from those several independent research studies, as the I-squared equal to 37% and the p-value greater than 0.05. I have inserted here a data for 10 studies that have an outcome that is continuous, and each study was arranged into the participant arranged into treatment group and control group, and the standardized mean difference was calculated, and the forest plot is generated as described here. This forest plot is produced from the SPSS software. It is based on the SMD and it is a continuous outcome. And this is another forest plot produced from our statistics, which is similar to the previous one. Again, it has 3 sections in it. This meta-analysis for continuous SMD. It has a section for descriptive, which shows the experimental and the control, the total, the mean, the standard deviation for each study, and for each group within each study, experimental and control, and the calculated effect size, which is the standardized mean difference, and the 95% confidence interval and the weight percentage. And the middle section here is diagrammatic representation for SMD with squares and diamond. And here at the third section is the overall effect size, which is the SMD and 0.72 with heterogeneity test is calculated, and the models used here is the random effects models. So this is the format the forest plot output will be produced. If the effect size is based on correlation coefficient, then the outcome is should be continuous, and this data here I had placed for 12 study. For each study the correlation coefficient is given with a total number of participants. A plot looks like forest plot for this correlation meta-analysis looks like this format. This is generated from R Studio, and again a section for descriptive, a section for diagrammatic, and a section for overall effect size and heterogeneity test. And the effect size here is the correlation coefficient that is calculated for each study with 95% confidence interval, and an overall effect size and overall correlation coefficient for all those 12 studies when they are combined with the 95% confidence interval. Another data has been inserted here based on an outcome that is proportion, in which the event happening in a group of individuals were counted and the total number of participants were also included. And the forest plot from proportional meta-analysis looks like this format, which is produced or a layout which is produced from the R Studio. Again, a descriptive section, diagrammatic section, and overall effect size. And the proportion here, or the effect size, is the proportion, as the proportion for each individual study is calculated with 95% confidence interval, and also an overall proportion from the combined 12 studies with 95% confidence interval. If the outcome is binary, which has 2 levels in it, and the effect size is odd ratio as shown here in this data set. Two groups, treatment and reference, and the event occurring in the treatment is counted and the event occurring in the reference group is counted. Then a forest plot can be generated from binary meta-analysis in a layout as shown here: descriptive, statistics, diagrammatic representation, and overall and heterogeneity. As shown for the binary outcome, the descriptive shows the event happening in the experimental and in control and the total of participants. The effect size is an odds ratio for each individual study is calculated with 95% confidence interval, and an overall estimate of the odds ratio also is given here. The required effect size is a risk ratio when the outcome is binary, as shown here, data from 12 studies that each study has two groups in it, intervention and control, and the event occurring is counted, event not occurring is counted, total number of participants counted, and the control, the event happening in the control is counted, not happening in the control is counted, and the total also is counted for the control. The output for the forest plot for this binary meta-analysis generated here. This is the format generated from an SPSS software. Again, 3 sections: descriptive, diagrammatic, and overall with heterogeneity, and risk ratio is calculated for each individual study with p-value 95% confidence interval, and the overall risk ratio for the 12 combined studies is given here with 95% confidence interval and a p-value, and the heterogeneity test for with I squared and Q value.